There is a quiet moment that comes somewhere in the sixth or seventh decade of life. It arrives differently for everyone. For some, it is the first time reaching for something on a high shelf and hesitating. Not because of the reach, but because of the uncertainty in the knees. For others, it is the morning when the mind feels slower, when the name of a familiar person hovers just out of reach, when the day feels heavier than it should before it has even begun. In that moment, a question forms, often unspoken, sometimes pushed aside. Is this simply what aging feels like? Is this the unavoidable price of having lived long enough to reach this point? Most people never ask the question out loud. They accept the slowing, the fading, the gradual retreat from full engagement with life as something inevitable. And in that acceptance, they overlook something essential. The body is not simply wearing down like an old machine running out of parts. It is responding, constantly and precisely, to the conditions in which it finds itself. The tiredness, the mental fog, the slower recovery. These are not commands from a pre-written script of decline. They are signals. They are the body's way of saying that something in the environment, something in the daily pattern, something in the accumulated weight of years and choices, has shifted the balance. What if the story you have been told about aging, the quiet assumption that decline is simply what happens after a certain point, is incomplete? What if the changes you notice are not the voice of time itself, but the echo of something you can actually influence? This is not a claim that aging can be stopped or reversed. It is an invitation to look deeper, to understand what is actually happening inside the body, and to discover that many of the forces shaping how you feel right now are not nearly as fixed as they appear. You are listening to the Longevity Library, where we archive the wisdom of healthy aging. The long view. Understanding the architecture of healthy aging. There comes a moment in life when the nature of time itself begins to feel different. The years accumulate, certainly, but something else shifts beneath the surface. The body speaks in a quieter voice, perhaps, or signals in ways that require more attention to interpret. For many people, this is the point at which questions about aging move from abstract curiosity to personal relevance. Not desperate questions, not fearful ones, but thoughtful questions about how the remaining years might unfold and what shape they might take. These questions are as old as human consciousness itself. Across cultures and centuries, people have wondered what determines the arc of a life. Why some individuals remain vigorous into their ninth decade, while others decline much earlier, and whether the way one lives can meaningfully influence these outcomes. The answers, as they emerge from decades of careful research, are neither simple nor simplistic. They do not reduce to a single pill or a single practice, but they do form a coherent picture, one that reveals aging not as a random process or an inevitable decline into frailty, but as something with its own internal logic, its own rhythms, and its own responsiveness to the conditions in which a person lives. This is not a story about defying aging or cheating time. It is a story about understanding the architecture of healthy years, the structures and patterns that support vitality across decades. It is a story about what happens inside the body at levels invisible to the naked eye, and how the choices made in daily life reach those hidden places. And it is, ultimately, a story of reassurance. Because what the research increasingly shows is that many of the most powerful influences on how we age are things we already encounter every day. Things we can learn to work with rather than against. The inner landscape of time. To understand how aging works, it helps to step away from the mirror and the calendar and look deeper, into the spaces where life operates at its most fundamental level. The body is not a single thing that ages uniformly. It is a collection of systems, tissues, and cells, each with its own rhythm of renewal and its own vulnerability to wear. The visible signs of aging. The gray hair and the lines on the face are merely the surface expressions of processes that have been unfolding for decades at microscopic scale. Inside nearly every cell, there is a nucleus containing the genetic material arranged in structures called chromosomes. At the ends of these chromosomes are specialized caps, repetitive sequences of DNA that serve a protective function. These caps are called telomeres, and they have become one of the most revealing windows into the biology of aging. Think of them as the buffers that keep the essential genetic information safe during the constant process of cell division. Every time a cell divides, which it must do to replace old or damaged cells throughout the body, these telomeres shorten slightly. They are, in effect, a kind of biological clock, counting down the divisions a cell has left. For many years, researchers understood that telomeres shorten with age, but they viewed this primarily as a marker, a sign that time had passed. More recent research has revealed something deeper. Telomeres are not merely passive indicators. They play an active role in how cells age. When telomeres become critically short, cells receive a signal to stop dividing. They enter a state called senescence, where they remain alive but no longer perform their functions or replenish themselves. This matters enormously for tissues that depend on ongoing renewal: the immune system, the lining of blood vessels, the skin, and even certain parts of the brain. The discovery of an enzyme called telomerase added another layer to this understanding. Telomerase has the ability to rebuild telomeres, adding back some of the length lost during cell division. It is not equally active in all cells. It operates more robustly in stem cells, the master cells responsible for generating new tissues, and at much lower levels in most ordinary cells. But its very existence suggests that telomere length is not simply a one-way slide toward shortness. Under the right conditions, the body has mechanisms that can support telomere maintenance, and potentially even lengthening. This is where the research intersects with daily life. If telomeres were purely determined by genetics, if their length at birth simply ran its course inexorably toward a predetermined end, then the conversation about aging would be very different. But study after study has shown that telomeres respond to the environment, to the conditions inside and outside the body. They shorten faster under certain circumstances and more slowly under others. They are, in the language of biology, plastic rather than fixed. The pace of aging within. One of the most striking findings from aging research is the degree of variation among people of the same chronological age. Two individuals born in the same year, having celebrated the same number of birthdays, can have vastly different biological ages when measured at the cellular level. Their telomeres may differ by hundreds of base pairs. Their patterns of gene expression may show different trajectories. Their risk profiles for age-related diseases may diverge significantly. This variation tells researchers something important. Chronological time, the steady tick of the calendar, is only one part of the story. Biological time moves at its own pace, influenced by a lifetime of experiences, exposures, and habits. Some people age in what might be called dog years, accumulating cellular wear at an accelerated rate. Others seem to move through time more gracefully, their cellular systems maintaining resilience decade after decade. Understanding why this happens requires looking at the forces that shape biological aging. These forces are many, and they interact in complex ways. But they can be grouped into categories that make the picture clearer. There are the influences that come from inside the body, the genetic inheritance passed down through generations. There are the influences that come from outside, the physical and social environment in which a person lives. And there are the influences that arise from behavior, the patterns of activity, rest, and nourishment that repeat day after day. Each of these categories matters, but the balance among them shifts over a lifetime. Genetic factors establish a range of possibilities, a kind of starting point. They influence how long telomeres are at birth, how efficiently cells repair damage, how robustly the immune system functions. But within that range, there is considerable room for movement. The environment and behavior can push biological age toward the younger end of the genetic range or toward the older end. This is not a matter of perfect control. No one can control everything, but it is a matter of meaningful influence. The weight of years on the body's systems. When researchers speak of aging biology, they are referring to specific mechanisms that can be observed and measured. Telomeres are one such mechanism. Mitochondria, the tiny structures within cells that generate energy, are another. As cells age, mitochondria become less efficient. They produce less energy for the same amount of fuel, and they leak more byproducts that can damage surrounding cellular structures. This decline in mitochondrial function appears in nearly all age-related conditions, from muscle weakness to cognitive changes. Inflammation represents another pathway through which aging expresses itself. The immune system, designed to respond aggressively to threats and then return to baseline, can become stuck in a state of low-grade activation. This chronic inflammation, sometimes called inflammaging, damages tissues over time and contributes to the development of cardiovascular disease, metabolic disorders, and neurodegenerative conditions. It is not the dramatic inflammation of an infected wound or an allergic reaction. It is quieter, more persistent, and in many ways more damaging because it operates beneath awareness. The epigenetic clock offers yet another window into aging. Epigenetics refers to the system of chemical marks on DNA that influence which genes are active in which cells. These marks change with age, and the pattern of change is consistent enough that researchers can use it to estimate biological age with considerable accuracy. Some people have epigenetic patterns that look younger than their chronological years. Others show accelerated epigenetic aging. These differences predict health outcomes decades later, suggesting that the pace of aging is established relatively early and maintained over time. What connects these various mechanisms is their responsiveness to the conditions of life. They are not rigidly programmed. They shift in response to nutrition, to physical activity, to psychological states, to sleep patterns, to the quality of relationships, and to the exposures encountered in the environment. This responsiveness is the foundation of any meaningful approach to supporting healthy aging. It means that the way one lives reaches all the way down to the cellular level, influencing the fundamental processes that determine how well the body functions over time. The exposome, life as a biological influence. The term exposome has entered the scientific vocabulary to describe the totality of environmental exposures a person experiences from conception onward. It includes the air breathed, the food eaten, the water drunk, the chemicals encountered, the noise heard, the light seen, and the social interactions experienced. It is a vast and complex concept, but it captures something essential about aging. The body does not exist in isolation. It is constantly exchanging materials and information with the world outside, and these exchanges leave lasting marks on biological systems. Some of the most concerning findings from aging research involve chemical exposures that are now widespread in modern life. Plastics contain compounds like bisphenol A that can interfere with hormone signaling. Pesticides and herbicides, including those used in conventional agriculture, have effects that extend beyond their intended targets. Heavy metals such as cadmium and lead accumulate in the body over time and show dose-related associations with telomere shortening. These substances are not exotic. They are present in ordinary environments, in food, in water, in household dust, in the air of cities and towns. Air pollution deserves particular attention because it is nearly universal in populated areas. Fine particulate matter, small enough to penetrate deep into the lungs and enter the bloodstream. Triggers inflammatory responses throughout the body. Studies have linked long-term exposure to air pollution with accelerated telomere shortening and increased risk of cardiovascular and respiratory diseases. The effect is not large enough to be visible in an individual over a short period, but over decades it accumulates meaningfully. The recognition that environmental exposures influence aging is not cause for alarm. It is cause for awareness. Some exposures can be reduced through individual choices, such as filtering drinking water, choosing foods grown with fewer pesticides, or using ventilation while cooking. Others require collective action, cleaner air standards, safer chemical regulations, and changes in how communities are designed. At the individual level, the goal is not to eliminate all exposures, which is impossible, but to tip the balance toward those that support health and away from those that undermine it. The body's response to chronic pressure. Psychological stress occupies a unique position in the study of aging because it is both external and internal. It arises from events and circumstances in the world, but it is experienced and processed entirely within the body and mind. The research on stress and aging has become increasingly sophisticated, and it reveals a pattern that is both sobering and hopeful. Not all stress is equal in its effects on the body. The ordinary pressures of daily life, the deadlines, the traffic, the minor frustrations, do not consistently predict accelerated aging. The body is designed to handle these short-term challenges, mounting a response and then returning to baseline. What matters more is chronic stress, the kind that goes on for years without relief. Caring for a spouse with dementia, living in an unsafe neighborhood, enduring an abusive relationship, facing persistent financial insecurity. These are the circumstances that leave marks on cellular aging. The mechanisms linking chronic stress to aging involve the endocrine system and the immune system. When the brain perceives a threat, it activates the hypothalamic pituitary adrenal axis, leading to the release of cortisol and other stress hormones. In the short term, this response is protective. It mobilizes energy and focuses attention. But when cortisol remains elevated for months or years, it begins to have damaging effects. It promotes inflammation, impairs immune function, and accelerates the shortening of telomeres. Rumination, the tendency to replay stressful events mentally and to anticipate future problems, extends the physiological stress response far beyond the actual stressful encounter. Someone who experiences a difficult interaction at work and then thinks about it for hours afterward, or wakes up in the morning already worrying about the day ahead, keeps the stress response activated continuously. This is why waking states are so revealing. The moment of awakening offers a kind of biological snapshot. People who wake up already anxious, already bracing for the day, show different cortisol patterns and different cellular markers than those who wake with a sense of calm or anticipation. Pessimism, as a general orientation toward life, has also been linked to shorter telomeres. This does not mean that optimism is a magic shield against aging. It means that the habitual patterns of thought, the lens through which events are interpreted, influence physiology. Recognizing one's own tendencies, whether toward pessimism, toward rumination, toward chronic worry, is the first step in loosening their grip. The goal is not to eliminate these tendencies entirely, which may not be possible, but to see them clearly and to reduce the time spent caught in them. Food as information for cells. The relationship between nutrition and aging has been studied from many angles. And while debates continue about specific details, the overall pattern is remarkably consistent. Whole foods, eaten in forms close to their natural state, support healthier aging. Highly processed foods, particularly those stripped of fiber and loaded with refined sugars and unhealthy fats, are associated with accelerated aging. Sugar-sweetened beverages have emerged as a particularly clear example of this pattern. Multiple studies have found that people who consume sugary sodas regularly have shorter telomeres than those who do not. The effect size is meaningful, equivalent in some studies to several years of additional aging. The mechanism likely involves the metabolic disruption caused by liquid sugar. When sugar is consumed in liquid form, it enters the bloodstream rapidly, causing a sharp spike in blood glucose. This triggers an insulin response, promotes oxidative stress, and activates inflammatory pathways. Over time, repeated spikes of this kind wear down cellular systems. The issue is compounded by the addictive nature of sugar. Brain imaging studies have shown that sugar activates dopamine pathways similar to those involved in drug addiction. This is not an exaggeration or a metaphor. The same neural circuits that evolved to reinforce survival behaviors can be hijacked by highly concentrated sugars, creating patterns of consumption that are difficult to change through willpower alone. This is why environmental changes, such as removing sugary drinks from schools and workplaces, can be more effective than individual resolve. When the default choice is a healthy one, people consume less sugar without having to resist temptation at every turn. Processed meats, including bacon, sausage, and deli meats, show similar associations with accelerated aging. These foods contain compounds that promote inflammation and oxidative stress, and they are consistently linked to higher rates of chronic disease in population studies. Replacing them with whole protein sources, such as fish, poultry, legumes, or plant-based proteins, shifts the balance in a healthier direction. Coffee, interestingly, shows the opposite pattern. Regular consumption of caffeinated coffee is associated with longer telomeres in multiple studies. Coffee contains hundreds of bioactive compounds, including polyphenol with antioxidant and anti-inflammatory properties. The effect appears to be dose-related, with higher consumption linked to greater benefit, though individual tolerance and preferences vary widely. Omega-3 fatty acids, found in fatty fish and in fish oil supplements, have also been linked to telomere maintenance. One study found that the actual levels of omega-3s achieved in the blood, rather than the dose taken, predicted whether telomeres lengthened or shortened over time. Higher blood levels of these fatty acids were associated with telomere lengthening, suggesting that achieving adequate omega-3 status may support cellular aging. Vitamin D, which functions more like a hormone than a vitamin, appears to have an optimal range for telomere health. Levels that are too low are associated with shorter telomeres, as are levels that are excessively high. The sweet spot, based on current research, lies roughly between 40 and 60 nanograms per milliliter in the blood. This is consistent with vitamin D's role in immune regulation and inflammation control, both of which influence cellular aging. The weight that matters. Body weight and aging have a complex relationship that is easy to misunderstand. It is clear from large population studies that higher body mass index is associated with shorter telomeres and higher rates of age-related disease. But weight itself may be a proxy for deeper processes. Metabolic health. The efficiency with which the body handles glucose and insulin may matter more than the number on the scale. It is possible to be lean and metabolically unhealthy, with poor insulin sensitivity and elevated blood sugar. It is also possible to carry extra weight while maintaining normal metabolic function, a pattern sometimes called metabolically healthy obesity. Research has shown that changes in metabolic status, improvements in insulin sensitivity and glucose regulation, predict positive changes in cellular health regardless of whether weight changes dramatically. This is why markers like hemoglobin A1C, which reflects average blood sugar over 3 months, provide valuable information. They capture the body's metabolic state in a way that weight alone cannot. Continuous glucose monitors, which are becoming more accessible, offer even more detailed insights. These devices can show how an individual's blood sugar responds to specific foods, revealing patterns that are highly personalized. Two people eating the same meal can have very different glucose responses based on their genetics, their microbiome, and their metabolic history. Fiber emerges as a consistently beneficial factor in glucose regulation. When fiber is present in a meal, it slows the digestion and absorption of carbohydrates, blunting the glucose spike that would otherwise occur. This is why whole fruits, with their fiber intact, affect blood sugar differently than fruit juices, which deliver sugar rapidly without the buffering effect of fiber. It is also why processed foods, from which fiber has been removed to improve texture and shelf life, are more metabolically disruptive than their whole food counterparts. Movement as cellular signal. Physical activity influences aging through multiple pathways, and the research consistently shows that even modest amounts of movement provide meaningful protection. The threshold for benefit is lower than many people assume. In studies of high-stress caregivers, those who were sedentary had shorter telomeres, but those who were active did not. And active in this context meant approximately 10 minutes per day of vigorous walking. This is encouraging because it makes movement accessible to nearly everyone. 10 minutes is not intimidating. It does not require special equipment, gym memberships, or athletic ability. It can be integrated into existing routines. A brisk walk to the mailbox and back. A loop around the block after meals. A few minutes of marching in place during television commercials. The key is consistency, not intensity. More structured exercise provides additional benefits. But the relationship between exercise intensity and telomere length is not linear. People who run marathons have longer telomeres than sedentary people, but they are not dramatically better off than those who run 3 times per week. Extreme exercise carries its own costs, including increased risk of injury and greater demands on recovery time. The optimal approach for most people is moderate, regular activity that can be sustained over years and decades. Exercise also helps break the cycle of rumination. Vigorous movement shifts attention away from repetitive thoughts and provides a physiological reset. The improved mood and reduced anxiety that follow exercise are not just psychological. They reflect changes in brain chemistry and nervous system activity that support recovery from stress. This is one reason why people who exercise regularly often report feeling more resilient in the face of life's challenges. The mind's role in cellular maintenance. The connection between mental states and cellular aging has been explored through studies of meditation, mindfulness, and other contemplative practices. A study of a 3-week residential meditation retreat found that participants showed telomere lengthening over this relatively short period. The effect was most pronounced among individuals who scored higher on measures of neuroticism, those with a tendency toward negative emotions. This suggests that practices which calm the mind may be particularly beneficial for people whose natural disposition keeps them in a state of heightened arousal. Mindfulness. The practice of paying attention to the present moment without judgment appears to work partly by reducing rumination. When attention is anchored in the present, it cannot simultaneously be caught in replaying the past or anticipating the future. This breaks the cycle of chronic stress activation and allows the body's repair systems to function more effectively. Not everyone connects with sitting meditation, and the research does not suggest that this is the only path to the same benefits. Yoga, Tai Chi, Qigong, and other movement-based practices combine physical activity with mental focus, offering a dual benefit. Even walking in nature, when done with attention to the surroundings rather than distraction, can induce a meditative state. The important thing is to find a practice that feels sustainable, something that can become a regular part of life rather than another obligation. Heart rate variability. which reflects the balance between the sympathetic and parasympathetic nervous systems, offers a window into these effects. Higher heart rate variability indicates greater flexibility in the nervous system, the ability to shift between states of activation and rest. Practices that promote relaxation, slow breathing, meditation, gentle movement, tend to increase heart rate variability over time. This is one measurable sign that the body is spending more time in repair mode and less in threat response. Sleep as foundational repair. Sleep has been called the third pillar of health, alongside nutrition and exercise, and for good reason. During sleep, the body performs essential maintenance that cannot happen while awake and alert. The brain clears metabolic waste products that accumulate during the day. Memories are consolidated and integrated. Cells throughout the body undergo repair processes. Hormones are regulated. The immune system recalibrates. Chronic sleep deprivation or poor sleep quality accelerates multiple aspects of aging. Telomeres shorten faster. Inflammation increases. Cognitive function declines. Metabolic regulation suffers. The relationship is bidirectional. Poor sleep accelerates aging, and the physiological changes of aging can disrupt sleep. But breaking the cycle is possible with attention to sleep habits. Regularity matters as much as duration. Going to bed and waking at roughly the same times, even on weekends, supports the body's internal clock. Exposure to natural light during the day, particularly in the morning, helps anchor this clock, dimming lights in the evening and reducing exposure to screens, which emit blue light that suppresses melatonin, signals to the brain that sleep is approaching. The sleep environment also influences quality. A cool, dark, quiet room supports the drop in body temperature that accompanies sleep onset. Comfortable bedding, minimal noise, and darkness all contribute. For those who struggle with sleep, addressing these fundamentals is the first step before considering more interventions. Social connection as biological need. Human beings evolved as social creatures, and this is reflected in physiology. The quality of social relationships predicts health outcomes with a consistency that rivals smoking, blood pressure, and cholesterol. People who feel supported and connected to others live longer, healthier lives than those who are isolated or in conflicted relationships. The mechanisms linking social connection to aging involve stress regulation. Supportive relationships buffer the effects of stress, providing resources for coping and a sense of safety that dampens threat responses. Conversely, loneliness and social isolation activate the same neural pathways as physical threat, keeping the body in a state of vigilance that accelerates aging. Positive relationships also provide meaning and purpose, which themselves are associated with better health outcomes. Having reasons to get up in the morning, people who count on you, projects that matter to you, roles that give structure to days. All of these support healthy aging. They keep people engaged with life and motivate the behaviors that maintain health. The quality of relationships matters more than quantity. A few deep connections provide more protection than many superficial ones. And relationships need not be with family or longtime friends. Community connections, volunteer roles, participation in groups based on shared interests, all can provide the sense of belonging that supports health. The prenatal origins of lifelong health. One of the most profound insights from aging research is that the trajectory of health is influenced by conditions experienced before birth. Telomere length at birth, which is shaped by genetics and by the prenatal environment, predicts telomere length decades later. Babies born with shorter telomeres have less buffer against subsequent wear and tear. Maternal stress during pregnancy has been linked to shorter telomeres in cord blood. Life events such as job loss, death of a loved one, or financial crises, when experienced by the mother during pregnancy, leave marks on the baby's cells. The same is true for stress experienced in the year before conception, suggesting that the health of both parents before pregnancy matters. Paternal health also influences offspring through epigenetic marks carried in sperm. Studies of obese men who underwent bariatric surgery showed that their sperm epigenetics changed significantly after weight loss, moving closer to the patterns seen in lean men. This demonstrates that lifestyle factors in men, not just women, influence the next generation's health. Socioeconomic factors, particularly education, also influence telomere length across generations. In a study of low-income pregnant women, those who had graduated from high school gave birth to babies with longer telomeres than those who had not, despite all participants being low-income. Education likely confers multiple advantages in terms of health literacy, access to resources, and ability to navigate complex systems, all of which translate to cellular benefits for the next generation. This research does not suggest that people are doomed by their beginnings. It suggests that the foundations of health are laid early, and that supporting the health of parents before conception and during pregnancy is one of the most powerful ways to influence population health across generations. For individuals, it offers perspective on the many influences that have shaped their own biology, and it underscores the importance of creating conditions that support the next generation. Sex, hormones, and the longevity gap. Women live longer than men in virtually every country and culture where data are collected. The gap is not trivial, amounting to several years on average. Understanding why has proven surprisingly complex, but research points to fundamental biological differences that are present from the beginning. Females are born with longer telomeres than males, a difference that persists throughout life. This is likely related to sex hormones, particularly estrogen, which has protective effects at the cellular level. In experimental models, estrogen upregulates telomerase and improves mitochondrial function. It enhances energy production while reducing oxidative stress, a combination that supports cellular longevity. The relationship between reproductive lifespan and aging provides additional clues. Women who experience menopause later and who give birth later in life tend to have longer telomeres. Longer exposure to endogenous hormones appears to be protective. However, hormone therapy in humans is complex, and the research does not support simple conclusions about hormone replacement. The relationship between hormones and aging is an active area of investigation with many unanswered questions. For men, the picture is different but equally complex. Testosterone has effects on multiple tissues, including the immune system, that influence aging trajectories. The health behaviors more common among men, such as higher rates of smoking, alcohol consumption, and risk-taking, contribute to the longevity gap but do not fully explain it. The 2X chromosomes that women carry may provide redundancy for certain genes, offering protection against mutations that would otherwise cause disease. The rhythm of days and years. Time is experienced in rhythms. The daily cycle of light and dark. The annual progression of seasons. The longer arc of decades. These rhythms are not merely external. They are built into biology through circadian clocks present in nearly every cell. Disruption of these rhythms, through shift work, jet lag, or irregular schedules, has measurable effects on health and aging. The master clock in the brain coordinates peripheral clocks throughout the body, synchronizing them with the external world. When this coordination is disrupted, cellular processes fall out of alignment. Digestion occurs at times when food is not present. Repair processes are activated during waking hours when they cannot proceed efficiently. Inflammation increases. Metabolic regulation suffers. Supporting circadian rhythms means living in ways that respect the body's built-in timing. Morning light exposure helps set the clock for the day. Regular meal times support metabolic rhythms. Consistent sleep and wake times allow repair processes to complete their cycles. These are not rigid requirements, but principles that can be adapted to individual circumstances. Even small adjustments toward greater regularity can improve how the body functions. The question of measurement. As interest in biological aging has grown, so has the availability of tests that claim to measure it. Companies now offer telomere testing directly to consumers, along with epigenetic clocks and other biomarkers. The appeal is understandable. The idea of knowing one's biological age, of having a number that reflects how well the body is aging, is compelling. But measurement at this level carries complexities that are easy to overlook. Telomere length measured in blood cells is an average that reflects the health of the immune system, not necessarily the health of other tissues. It can fluctuate with recent infections, with stress, with technical factors in the laboratory. A single measurement provides limited information, and dramatic changes between measurements are as likely to reflect measurement error as true biological change. The interpretation of results is also challenging. In population studies, shorter telomeres are associated with higher disease risk, but this does not translate directly to individual prediction. Someone with short telomeres may live a long, healthy life if other factors are favorable. Someone with long telomeres may develop disease for reasons unrelated to telomere length. The statistical associations that hold across large groups do not determine any single person's outcome. For those who choose to pursue testing, the most useful approach is to establish a baseline and then monitor changes over time in response to lifestyle modifications. This shifts the focus from a single number to the trajectory of change, which is more informative. It also requires understanding the limitations of the tests and the potential for psychological reactions to unexpected results. The integration of influences. The factors that influence aging do not operate in isolation. They interact, amplify, and sometimes cancel each other. A healthy diet matters more when combined with regular physical activity. Stress management supports better sleep, which in turn supports metabolic health. Social connections provide motivation for healthy behaviors and buffer the effects of life's challenges. This integration is important because it means that small changes in multiple areas can add up to meaningful differences. Improving sleep by 30 minutes. Adding a 10-minute walk to the daily routine. Replacing one sugary drink with water. Calling a friend once a week. These are not dramatic interventions, but over years and decades, their cumulative effect on cellular aging can be significant. The research does not support the idea of a single magic bullet for healthy aging. No pill, no supplement, no single practice has been shown to override the effects of a lifetime of unhealthy patterns. What the research supports is a comprehensive approach, one that addresses the multiple pathways through which aging proceeds and that recognizes the interconnectedness of body systems. This is reassuring rather than discouraging. It means that there are many points of entry, many places where positive changes can begin. Someone who has never exercised can start walking. Someone who eats a poor diet can improve one meal at a time. Someone who feels isolated can reach out to one person. Each small change creates conditions that make other changes more likely, building momentum over time. The perspective of decades. Aging is not an event, but a process, one that unfolds over decades. The choices made in midlife predict health outcomes in later life with remarkable accuracy. This lag time, the gap between cause and effect, makes it easy to underestimate the importance of daily habits. The effects are not visible immediately. They accumulate silently until, decades later, they manifest as disease or vitality. This is why consistency matters more than intensity. A moderate habit sustained for 30 years has more impact than an intense effort abandoned after 3 months. The body responds to the long-term average, not to occasional bursts. This is also why it is never too late to make changes. Even in later years, improvements in nutrition, activity, and stress management produce measurable benefits. The trajectory can shift at any point. The goal is not perfection. No one maintains optimal habits every day, every week, every year. The goal is a general direction, a tendency toward patterns that support health rather than undermine it. Occasional deviations are normal and do not derail the overall trajectory. What matters is the long-term average, the accumulated weight of countless small decisions made over years, the architecture of healthy years. Stepping back from the details, a coherent picture emerges. Healthy aging is supported by patterns that reduce chronic stress on the body, provide adequate resources for repair and maintenance, and maintain flexibility in physiological systems. These patterns include eating whole foods, moving regularly, sleeping sufficiently, managing stress, maintaining social connections, and reducing exposure to environmental toxins. None of these patterns is new or surprising. They have been recognized across cultures and centuries as the foundations of health. What research adds is the understanding of how they work at the cellular level, how they reach the telomeres and mitochondria and epigenetic marks that determine the pace of aging. This understanding does not change the recommendations, but it deepens the reasons for following them. It also provides a kind of reassurance. The factors that most influence how we age are, to a significant degree, within our influence. They are not mysterious or inaccessible. They are the ordinary stuff of daily life. The food we eat, the way we move, the people we connect with, the rest we take, the thoughts we think. These are things we encounter every day, things we can learn to work with rather than against. The long view of aging reveals that time is not simply a force that acts upon us. It is something we participate in, something we shape through the conditions we create in our bodies and our lives. The years accumulate, certainly, but how they accumulate, whether they build toward vitality or decline, is influenced by patterns that repeat day after day. This is the architecture of healthy aging. Not a blueprint for defying time, but a way of living well within it, of making the most of the years we have. If there is a single thread that runs through everything we have explored, it is this. The body is not a passive passenger riding out the years. It is listening, constantly, to everything you do, everything you eat, everything you think, every relationship you tend, every rest you take. It is responding not to the calendar, but to the conditions you create. The question is not whether you can stop time. The question is whether you can create conditions that allow your body to do what it already knows how to do. To maintain, to repair, to renew, for as long as possible. Take what you have heard here and hold it gently. Let it sit in the background of your days. Notice the small moments, the choices that appear insignificant, and consider what they might be telling your body about the life you want to live. If something in this conversation stirred a thought, a memory, a question of your own, we would be honored to hear it in the comments. And if you would like to continue exploring the wisdom of healthy aging, you will find more conversations like this one waiting for you in the library. Subscribe if you do not want to miss them. This is the Longevity Library. Listen, learn, and live longer.